Now that we've been in the forest for a while, I'm going to take you back to the laboratory. And I'm going to primarily discuss, it's working good. I'm going to primarily discuss two different studies or areas that we're working on right now. One of those was research conducted by Dr. Janita Hajiabdik Gary, and she's our genetics specialist, which I am not. And anyway, she has done some work looking at the genetic differentiation and spatial structure of Geosmithia morbida, of course, the causal agent of thousand canker disease. The point of this study was to evaluate the genetic composition of Geosmithia morbida populations within the native range of black walnut. And as you can see from this graphic, we have samples represented by the little purple circles are where we have confirmed isolates of Geosmithia, where they were collected. Those are mostly in Tennessee, of course, and there is also the isolate in North Carolina that Greg just talked about at the Big Creek. And there was also samples that we were sent from Oregon by Jay Scheid, if I'm not mistaken. And then, of course, there's also the sample that was found in Pennsylvania. We used a total of 62 confirmed isolates from these areas. The DNA was extracted and processed using the standard protocols, which I'm not going to go into at this point. And basically what we found was there seemed, according to a program called Structure, which makes these phylogenetic trees, there seemed to be two different populations. One, this large red area, these are all of the Tennessee isolates from our counties, Knox County, Blount County, Anderson, Loudoun, and Sevier were included in this study. And basically the genetic population here is all related. And the sample that was found in North Carolina and also the Oregon seemed not as related as the Tennessees were among themselves. Now, this doesn't necessarily mean that the Geosmithia we isolated in North Carolina came from Oregon. It just means that neither one of those, the one from Great Smoky Mountains National Park or the population from Oregon, are as related to our Tennessee population. And the major reason we think that the Oregon and the North Carolina fell out together is because we just don't have enough samples from the West. And we've had a little difficulty in getting samples there. If you'd like more information, this is a published article. It's in Current Genetics, and we can hook you up with this reference. But once we found this information and there was not a lot of Western samples, we decided that maybe we should go get some. So Dr. Gary and Catherine Nix, who works in Paris Lambdin's lab, went out west and decided to collect as much as they could. And of course, they were helped out by Whitney Cranshaw, Ned Tisserat, and I believe Rachel Fithian. And they directed them to sites that they knew were infested. And there were a total of 68 samples of wood taken, and you can see the species listed here from each of these states. Whitney and Ned participated and helped us in all of these states except for Oregon, and the samples that we were given, the samples from Oregon were sent by Jay Scheid at Oregon State University. So we started looking at these. The things that really jumped out at us were things that were different than what we normally see. I've worked in Dr. Wyndham's lab for quite some time, and I've been involved with our studies in Geosmithia since they were first discovered in 2010. And I've received all the samples that have come in for testing, and I've never seen samples that were so loaded with galleries and cankers. We just don't see that in the samples from this part of the country. There were cankers on young wood, cankers on old wood, and they were ubiquitous. And I really like this slide. It's not quite as dramatic as some of the others, but when you look at this, you really see where the name thousand canker disease came from, because all of these tiny little dots are actually individual cankers. We also found Geosmithia morbida actually growing inside the galleries, which is something we do not normally see in eastern samples. We found galleries that contain beetles, with Geosmithia growing both in the gallery on the wood itself and also on the beetle itself. As you can see from this picture, the fungus is growing directly out of the beetle. So what do we do with all this stuff? The easiest method for isolating Geosmithia out of the wood is to place the wood chips containing cankers in a moist chamber. And a moist chamber is simply a petri dish with some moistened filter paper, and you put the lid on, you let it incubate for 3 to 4 days, and examine the wood under a dissecting scope at low magnification, and voila, you have little conidiophores sticking up all over the place. You can then harvest these and grow them out on the medium. Another method is what we call direct plating, and that's where instead of placing the chip of wood with the canker in a moist chamber, you just put it directly onto medium. The medium that we use is a basic potato dextrose agar. It's readily available commercially, and we use a very low strength, about a 1/10 strength. And the reason being is that this wood contains a lot of other fungi. It's not just the Geosmithia world. And Geosmithia tends to grow a little slower than, say, Fusarium or Bacillomyces, some of the other fungi that are present, and it overwhelms the Geosmithia. And we think we've probably had some positive samples that we could not identify simply because our Geosmithia was overgrown by other fungi. And this is just a close-up because I like this picture with the cankers. It's very clear. And this is directly plated. We can also, as far as the direct plated method goes, we can grab some of that, the Geosmithia that we find in the galleries. I've also placed beetles on medium. And again, you get cultures, but a lot of times they are very overgrown with other fungi, and they're very difficult to isolate and clean up. You tend to do repeated transfers, and it can be very time consuming, which means money. This is just an example of direct plated where we've taken fungus out of the gallery and just made a smear in the medium, and then the fungus grows out of that. This white area is where the conidiophores have developed, and you'll get your spore production. So once we have all these cultures that we've initiated, you know, you've got several hundred plates sitting there on the counter. What are you going to do with all these? Well, first of all, we have to go in and throw out things that are obviously not Geosmithia, and there are a lot of those. Then we can look at the remaining cultures and look at those under the microscope, take samples, and look to confirm morphologically that they are Geosmithia. One thing I would like to point out, these are all confirmed Geosmithia cultures, and I'd just like to mention the diversity here. We have some that grow completely under the medium, and there's very little to no spore development. We have others that, you know, they're kind of a mix. Part of it's growing under the surface and some is on top. And then you have others that are predominantly on top, and that's where you really get a lot of spore development. As you can see, it's much, much wider. But there is the common structure where you have an introduction point and then they kind of radiate out. And that's something we notice in cultures that we later confirm. And as I said, we confirm morphologically with microscope. One of the things we look for is the hyaline style, which means basically clear, nice. There's not a lot of other structure in that hyphae. And we've actually, being the geeks we are, sat around one day and measured those, and it seems that Geosmithia is very regular in that. Each septa is very regularly spaced. Of course, the defining characteristic is this conidiiform structure. It's almost like a hand with a thumb sticking out. We notice that, and the conidia are also in chains. And most of the time when you make a microscope slide, you bust those chains apart. So I was really pleased to find a chain structure in here. The shape of the spore itself is also very indicative of Geosmithia. So what does all this mean? It all comes down to a graph. We started out, the direct plating method is in the kind of orange color, and the lavender color is our moist chambers. We started out with over 400 cultures, direct plating, and over 191 in the moist chambers. We had those cultures that were obviously not Geosmithia, so we tossed those out. And that was almost half of the direct plated cultures that we had. We later looked at those that were possibly Geosmithia, so once we've gotten rid of the ones we know definitely are not, we had about 204 of the possible Geosmithia and 163 from moist chambers. When we later looked at those and confirmed morphologically what were and were not Geosmithia, we ended up with 80 cultures that were derived from direct plating and 86 that were derived from the moist chambers. And as you can see, that's a 19% success rate and a 45% success rate. So looking at this graph, it's obvious that as far as Western samples are concerned, the method of preference is using a moist chamber situation and pulling those cultures off of wood that is developed in a moist chamber. So what does this mean? Well, basically for us, it means fewer supplies. You're not having to produce as much media. You're not having to do so many transfers. You can just put it in this little petri dish, let it grow up, and then transfer it and onto the more costly medium. So if you're not making medium, you're not doing these transfers, of course that's less labor, and many times that's the most expensive component in a research project. This also means a faster turnaround. So if you're inundated with samples from the field, you know that perhaps if you use this moist chamber method that you can process these samples a lot quicker. And overall, that's a lower cost. And that's the bottom line with everybody in research, is to try to get as much information as you can with the least expenditure as possible. So that, all of that being said, it takes a lot of people to do this, and we are grateful to Whitney, Ned, and Rachel for their assistance in collecting samples, and especially to Jay Scheid because he sent us all of the organ samples that we had. And then, of course, there's project members here. If it takes a village to raise a child, it takes a nation to conduct research nowadays. So we have all of these people, especially Paul Merton from Forest Service and Muncie from TVA. And then we have other students and collaborators here that aren't officially a part of the project, but that make it happen. And of course, we would like to thank our funding sources, the USDA ARS, the USDA Forest Service, and of course the Wood Tiger Fund.